transport that requires energy. So our first three mechanisms did not require energy. They were spontaneous or downhill. And then active transport, we know, requires energy. So it's going to need some type of pump or some type of other molecule that is moving according to its gradient to donate or to liberate some energy for the movement of these ions actively. So typically we have ions or molecules or substances that are moving against their concentration gradient, okay? So think about sodium as a really good example. We have sodium at high concentrations outside the cell and low concentrations inside the cell. And so to move sodium out of the cell, we need active transport. We need some type of pump or some type of energy donating process to facilitate that movement. Okay, so again, looking at this example here, we have side A and side B. We have an active transporter. Now we have equal concentration of particles on either side. And so we have this pump that is helping to create this steady state where we're pumping, in the example that we'll look at, sodium against its concentration gradient to help it build up in excess on side B. This is how we get excess sodium outside the cell. We have that sodium-potassium pump, which we're going to speak a lot about, and it is moving sodium against its concentration gradient. This is how we get to keep sodium in excess outside the cell. Otherwise, if we didn't have that active pump, sodium would dissociate to its equilibrium. It would dissociate till it is at equal concentrations on either side, which we don't want in the case of excitable cells. Okay, so the characteristics of a pump is that it is a membrane protein. Again, so another one of our integral protein examples. So it's fixed in the membrane permanently. It functions as a transporter and an enzyme. So it also has this enzymatic component to it. So the enzymatic component is where we can use the energy from ATP, right? We can liberate that energy, take that phosphate off, and use that energy to actually facilitate the movement of these ions. So this is where the energetic demands are met through the use of this enzymatic portion of the pump. So we can facilitate or harness some energy here to do work. And this also has that specificity. So for example, a sodium-potassium pump will only bind sodium and potassium, and it can also demonstrate saturation. So if all those sodium sites are bound up and all those potassium sites are bound up, then we have the limiting transport of these ions because we have that limiting resource. We only have a set number of sodium-potassium pumps. Okay, so that's really important to point out. So far, we've seen that with three of our mechanisms. We've seen that with active transport, we've seen that with facilitated diffusion, and we see that with channels, right? If there is saturation of our carriers, then we have a drop in the rate of transport. Okay. Now, primary active transport is a little bit different from secondary active. Primary active transport, the energy that we need comes from that ATP hydrolysis, right? So the sodium-potassium pump is an example of this. Okay, so we said that we need energy, and the difference between primary and secondary active transport is that the energy source is different for both of these mechanisms. So for primary active transport, the energy comes from ATP. All right, looking at the sodium-potassium pump as an example, we bind 3 sodiums, they get pumped out of the cell. We bind 2 potassiums, they get pumped into the cell. And the hydrolysis of ATP is driving the energetic needs for this process. Secondary active transport is slightly different. We still need energy, but the energy here comes from somewhere else. So we spoke about passive movement. So during passive movement, molecules are moving according to their concentration gradient. Sodium is an example. So sodium is at high concentrations outside the cell and low concentrations inside the cell. So when sodium moves into the cell, it's doing so according to or down its concentration gradient. That is a mechanism of passive movement or passive transport. And so that donates or liberates some energy, and we can see that by this green arrow. Now, the energy that donates can be used by this carrier, this pump, to drive the movement of glucose against its concentration gradient. Again, we have glucose at a high concentration outside the cell, right, usually in the blood, and a low concentration inside the cell. And so to move glucose inside the cell, it's going to be moved against its concentration gradient, okay? So moving glucose down against or up its concentration gradient requires some energy that we can use from the energy that is liberated from sodium, which moves down its concentration gradient. We typically call this type of secondary active transport co-transport because we have two molecules moving in the same direction. One is moving down its gradient, one is moving against its gradient, and they're basically using the energy that the other one is donating. Another type of, or rather the example that we're speaking about here is the sodium-linked glucose pump. So this is just to expound some more on this co-transport. The specific pump that's here is called sodium-linked glucose pump, and this is facilitating that secondary active transport. Diffusion of sodium provides the energy for the diffusion of glucose. Another type of example here is the counter transport, and this is seen with the sodium-linked proton pump. So alternatively to the movement of ions in the same direction, we have the movement of ions in opposite directions here. So it's still secondary, it's still active because it requires energy, but we have movement of molecules or ions in opposite directions. So we're still borrowing or using that energy that sodium liberates, but we're moving protons or hydrogen ions in the opposite direction. Okay? And this is called counter transport, which is another example of secondary active transport. Okay, any questions so far? Any questions so far? All right, now this is a really nice study table. It's going to help you distinguish between these mechanisms of transport. And as you can see here on the left, we have the two types or the three types of transport that are passive. So these are spontaneous or down the concentration gradient. And on the right, we have the two types of transport that require some type of energy, okay? We have simple diffusion. We know simple diffusion doesn't require anything. It's usually oxygen or fat-soluble molecules, carbon dioxide, moving into the cell or out of the cell according to the concentration gradient. They do not require any type of help or carrier in any way. And then we have channels, which are those pores we talked about. They can facilitate the movement of water or ions. We have a carrier or facilitated diffusion, which is usually moving some type of organic molecule. Glucose is the example that we looked at. And then as far as our active transport goes, we know that both of these require some type of energy, but the energy source is different. So for primary active transport, the energy source is the hydrolysis of ATP. Usually the sodium-potassium pump is a really good example of this. And then for secondary active transport, we're using the electrochemical gradient of another molecule. So something else is moving according to its gradient, and we use the energy that's liberated there to move organic molecules or ions against their concentration gradient. Okay. Now I want to point out the coordination of all of this. So although we're speaking about active and passive transport kind of exclusively, right, we're talking about them differently. On a single cell, we have a very nice coordination of all these mechanisms to help bring about our cellular function. So we can see that there's a sodium-potassium pump here. Okay, it's pumping sodium out and it's pumping potassium in. But on the other side of the membrane, we can also see some of our leak channels. Okay, the leak channels are allowing sodium to leak into the cell according to its gradient, right? And potassium to leak out of the cell according to its gradient. So the concert, the coordination basically, right, the pump along with the channels acting in concert help to maintain the relative ion concentrations. This is why in the extracellular fluid we see more sodium, because of the coordination of these two types of transport mechanisms. And this is why also in the extracellular fluid we see less potassium. Okay, we have potassium building up inside the cell, and we have sodium building up on the outside of the cell because of the coordination of these two mechanisms.